Greetings to everyone watching this video. In this video, we will be learning about the process of translation and the Lac Operon. But before we move on to the main concept, let us know more about us at reWise Me. At reWise Me, you get 100 plus free quizzes, multiple play modes, leaderboard, exciting daily rewards, and weekly GK updates. Also, we have launched a mega quiz feature this month. You can stay tuned to our Insta page to know the details. Now, let us move on to the main concept. So when we talk about translation basically होता क्या है? हमने ये चीज पढ़ी है DNA, RNA, protein. So DNA to RNA to protein. We know that this step is known as replication. DNA to DNA. DNA to RNA is known as transcription. It is known as transcription. And RNA to protein is known as translation. So the process of conversion of mRNA to protein is known as translation. And translation कैसे होती है? It is defined by the sequence of bases on the mRNA. It is defined by the sequence of bases on the mRNA. अब हमें ये बात पता होनी चाहिए कि जो अमीनो एसिड्स हैं, now in the entire video I will be representing amino acids as AA with a circle. So हमें ये पता होना चाहिए कि जो amino acids हैं, they are linked together by a peptide bond. Yes, so the amino acids are linked together by a peptide bond. So in the first step of translation, what happens is charging of tRNA. Charging of the tRNA. Charging of the tRNA, or another name given to it, is the amino acylation of tRNA occurs. Amino acylation of tRNA. Now what is this charging or amino acylation? Basically when two tRNA, suppose this is a tRNA plus this is a tRNA, when they both of them come close together, a peptide bond is formed energetically. A peptide bond is formed spontaneously and energetically. Now this is known as amino acylation of tRNA. Now when we are talking about translation, we need to know about ribosome. So what exactly is ribosome? Ribosome is the cellular factory of proteins. Ribosome is the cellular factory of protein. So in ribosome, in a resting state, ribosome is composed of two subunits. One is the large subunit of the ribosome, and the other is the small subunit of the ribosome. The small subunit of the ribosome. Ribosome is responsible for protein synthesis. And what does ribosome actually contain? It contains structural RNAs, and 80 different types of proteins. It contains 80 different types of proteins. It contains 80 different types of proteins. Now, in the process of translation, when a mRNA encounters the small subunit of the ribosome, the process of translation begins. The process of translation begins. Now we need to know that the large subunit of the ribosome, it has this large subunit of the ribosome has two sites. It has two sites for amino acids to bind through it. And when the amino acids will be bound through it, it will be an energetic process. It will be an energetic process in ATP formation as well. It will be an energetic process. Now we need to know that the ribosome also acts as a catalyst. The ribosome acts as a catalyst as well, This is a very important statement. It has already been asked in many times in the NEET exam, that ribozyme which acts as a catalyst, it is the 23 rRNA in bacteria. This is a very important statement for you to note. It has been asked many times. Now, as you can see over here, this is the small subunit of the ribosome. This is the large subunit. Now, the sequence of bases, as you can see, the sequence of bases are decided by the bases on the mRNA. and on one side the tRNA brings the bases here, one side it brings the bases here, and when the bases are attached to their cognate tRNA, this growing polypeptide chain is grown, and this is the ultimate process of translation, right? Now we need to know, when we talk about an mRNA, suppose this is an mRNA, so we need to know that the mRNA, suppose from here to here, it has these non-coding regions, the mRNA, this part and this part. This part is collectively referred to as the non-coding region, the non-coding region of the mRNA, and this region is known as the untranslated region, untranslated region, or the UTR, UTR. ये बोथ 5' एंड पे भी होता है और 3' एंड पे भी होता है। इतने पार्ट में कोई एक्टिविटी नहीं होती, ये बस अनट्रांसलेटेड रीजन्स हैं, ये बोथ स्टार्टिंग स्टॉप पे एक्स्ट्रा प्रेजेंट होते हैं। We need to know for the initiation process of translation, initiation process के लिए जो mRNA होता है वो पहले AUG को रिकॉग्नाइज करता है, which all of us know is the start codon. अब AUG जो है, it is further recognized by the initiator tRNA. It is recognized by the initiator tRNA. We need to note a very important point, that there are no tRNAs for stop codons. There are no tRNAs for stop codons. Stop codons के लिए कोई भी tRNAs नहीं होते। अब होता क्या है, this was the initiation phase, the first phase, the initiation phase. Next phase में जो translation होता है, it enters the elongation phase, जहाँ पे वो elongate करता है। It enters the elongation phase, जो हमने previous slide में देखा था, ऐसे ये growing polypeptide chain. This phase is known as the elongation phase. ये हो रही होती है elongation phase of the protein. Ultimately, the third and the last phase known as the termination phase. Termination phase में क्या होता है, a release factor, a release factor, a release factor comes and it stops the translation. It stops the process of translation. Stops the process of translation. Up we need to learn about the Lac Operon. So first we need to know who elucidated the Lac Operon. Lac Operon को ढूंढा किसने? One was a geneticist and the other was a biochemist. So the geneticist who worked in the, who contributed to the Lac Operon, the geneticist was Jacob, while the biochemist was Monod. These two people are responsible for the discovery of Lac Operon. अब Lac Operon का definition क्या है? Lac Operon is mainly defined as a, the precise definition of Lac Operon is, it is a polycistronic, it is a polycistronic structural gene, polycistronic structural gene which is regulated, a polycistronic structural gene which is regulated by a single common promoter, by a single common promoter. It is regulated by a single common promoter and a regulatory gene, and a regulatory gene. We need to know that this type of these operons, they are common in bacteria. They are common in bacteria and they are of various types. Now, when we talk about the operon in our syllabus, which is the Lac Operon, here, Lac nothing is but lactose. Lac is nothing but the lactose. Now, the Lac Operon, it consists of one regulatory gene. The Lac Operon consists of one regulatory gene. Lac Operon पे एक regulatory gene होती है, which is the I gene. I gene. मैं अभी आपको पूरा Lac Operon बना के दिखाऊंगा. It is the I gene. एक regulatory gene होती है and it has three structural genes. It has three structural genes. These genes are Lac Z, Lac Y and Lac A. What do these code for? The Lac Z codes for beta galactosidase, Lac Y codes for permease, and Lac A codes for transacetylase. ठीक है? The Lac Z codes for beta galactosidase, Lac Y codes for permease, and Lac A codes for transacetylase. हमें ये बात पता होनी चाहिए कि जो लैक ओपेरॉन है, लैक ओपेरॉन में जो हमारा सबस्ट्रेट होता है, द सबस्ट्रेट इज लैक्टोज। लैक ओपेरॉन में हमारा जो सबस्ट्रेट है दैट इज लैक्टोज, एंड क्योंकि वो सबस्ट्रेट है इट इज कॉल्ड द इंड्यूसर। अब क्योंकि वो सबस्ट्रेट है it is called the inducer. Now we have a trick to remember something. It is lion. I'll explain you what it is. Lion means L for lactose turns the inducer on. ON is combinedly on. Now what does this mean? I'll just explain it to you in the upcoming slides. Now one thing we need to note that if the lactose is provided in the absence of carbon, it enters the cells, through the action of permease, Just remember this statement. This statement, specifically this line, has been asked many times. So you need to remember this statement. So it enters in the action of permease. अब जो हमारा लैक ओपेरॉन है, डोंट वरी, आई विल जस्ट ड्रॉ द लैक ओपेरॉन इन द अपकमिंग स्लाइड। जो हमारा लैक ओपेरॉन है उसमें जो रिप्रेसर है, द रिप्रेसर इज कॉन्स्टिट्यूटेड एंड सिंथेसाइज्ड ऑल टाइम फ्रॉम द आई जीन। तो जो हमारा आई जीन है इट कोड्स फ्रॉम द रिप्रेसर। अब ये जो रिप्रेसर होता है, द रिप्रेसर बाइंड्स टू द रिप्रेसर जो है हमारा, द रिप्रेसर बाइंड्स टू द ऑपरेटर रीजन। ये जो है it binds to the operator region, जिसकी वजह से it stops translation. तो ये binds to the operator region and it stops translation. अब क्या होता है, let's know. लेकिन we need to know when the cell has lactose present. हमने अभी देखा था LION. So lactose turns the inducer on. So when lactose is present, it acts as an inducer. So when it acts as the inducer, what happens? It binds to the lactose binds to the repressor protein. उससे क्या होता है? It stops it from going to the operator region, from hindering the operator region. तो अब वो ऑपरेटर रीजन पे नहीं जा पाता, तो जब वो ऑपरेटर रीजन पे नहीं जा पाता तो प्रॉपर ट्रांसक्रिप्शन एंड ट्रांसलेशन अकर्स. सो ट्रांसक्रिप्शन एंड ट्रांसलेशन अकर्स. नाउ लेट अस सी द लैक ओपेरॉन ऑन द नेक्स्ट स्लाइड. अब हम देख सकते हैं कि ये लैक ओपेरॉन हमारे स्क्रीन पे ड्रॉन है. ये है the sequence of structural genes, that is PIPOZYA. The way to remember it is Piposia. So when you say Piposia, you remember the entire sequence of the Lac Operon. So अब क्या होता है? This repressor protein in the absence of lactose, when I tell you when lactose absent है, जब lactose absent है, that means inducer is off. लैक्टोज एब्सेंट है तो इंड्यूसर ऑफ है. सो क्या होता है जब लैक्टोज नहीं होता? सो दिस रिप्रेसर प्रोटीन गोज एंड बाइंड्स टू द ऑपरेटर रीजन, राइट? ये ऑपरेटर रीजन पे चला जाएगा रिप्रेसर प्रोटीन. तो जब ये रिप्रेसर प्रोटीन यहाँ पे आ गया और यहाँ पे इसने हिंडर कर लिया, तो अब हमारा जो Z, Y और A इनका ट्रांसलेशन नहीं हो सकता. तो फिर हमारे जो ये प्रोडक्ट्स हैं बीटा गैलेक्टोसाइडेज, परमीएज और ट्रांसएसिटाइलेज, ये नहीं बन सकते. लेकिन व्हेन लैक्टोज इज प्रेजेंट, lactose is present, what happens is lactose present है यानी inducer है. तो जब inducer है, so the inducer will go bind to the repressor protein like this. So when the inducer binds to the repressor protein like this, it cannot go and bind to the operator region. और जब वो operator region पे bind नहीं हो सकता तो इनकी proper translation होगी और ये वाले जो हमारे products हैं ये पूरी तरीके से बन जाएंगे. So I hope you understood the Lac Operon. सेम एग्जैक्टली सेम डायग्राम में बनाया है. ये रिप्रेसर है, ये रीजन आरएनए पॉलीमरेज फ्रॉम ट्रांसक्राइबिंग द ओपेरॉन. जब इंड्यूसर नहीं है तो ये रिप्रेसर यहाँ ऑपरेटर पे बैठ जाएगा जिसकी वजह से जेड वाई और ए नहीं हो सकते. लेकिन आप देख रहे हो जैसे ही इंड्यूसर आया तो ये इंड्यूसर बाइंड्स टू द रिप्रेसर प्रोटीन. अब जब वो रिप्रेसर पे बाइंड हो गया तो ये इनएक्टिव हो गया, तो दिस इज इनएक्टिव नाउ. तो अब ये ऑपरेटर पे नहीं जा सकता और जब ये ऑपरेटर पे नहीं जा सकता तो Z, Y और A प्रॉपरली ट्रांसलेट होंगे. अब हमें ये पता होना चाहिए कि जो इसके इंड्यूसर्स हैं, इंड्यूसर्स ऑफ लैक ओपेरॉन, इंड्यूसर्स ऑफ लैक ओपेरॉन are lactose and allolactose. Lactose and allolactose. They are lactose and allolactose. The last point in the Lac Operon which is to be remembered is Lac Operon is under the control of both positive and negative regulation. So the Lac Operon is under the control of both positive and negative regulation. So I really hope you understood the concept of translation and Lac Operon. I would request you to go and attempt the quiz on our app pertaining to this topic to test your learning pertaining to this concept. Thank you and have a nice day.